Good morning. Welcome back, everybody, to my daily lectures. I'm Dr. Bobby. On behalf of New Age Cosmetic White Edtech Healthcare Consultancy, I welcome all examination goers who are preparing for the qualifiers of the NEET exams, all in the Institute of Medical Sciences, the Council of Scientific and Industrial Research, conducting national eligibility tests for those appearing for the life sciences. Today we are at the topic of molecular biology of the cell, which is Unit 13 of the prescribed syllabus of CSIR-NET. It encompasses all details required for the other exams as well. So in the subject of molecular biology, we have methods in biology, details on the details of molecular biology and recombinant DNA methods. So we have a general recombination note. So we'd like to give all credits to the National Library of Medicine, the National Institute of Health, at the National Center for Biotechnology Information. Courtesy to them for the details provided to us to study for all students. And starting now with general recombination. In the two preceding sessions, or portions that were already covered, there's been discussions of the mechanisms that allow the DNA sequence in cells to be maintained from generation to generation with very little change. However, it is clear that these DNA sequences are occasionally rearranged. The particular combination of genes present in any individual genome, as well as the timing and level of expression of these genes, is often altered by such DNA rearrangements. In a population, this type of genetic variation is crucial to allow organisms to evolve in response to a changing environment. The DNA rearrangements are caused by a set of mechanisms that are collectively called genetic recombination. Two broad classes are commonly recognized: general recombination and site-specific recombination. In this part of the chapter, we discuss the first of these two mechanisms, and the next we consider the second mechanism. So in general recombination, also known as homologous recombination, genetic exchange takes place between a pair of homologous DNA sequences. These are usually located in two copies of the same chromosome, although other types of DNA molecules that share the same nucleotide sequence can also participate. The general recombination reaction is essential for every proliferating cell because accidents occur during nearly every round of DNA replication that interrupt the replication fork and require general recombination mechanisms to repair. The details of the intimate interplay between replication and recombination are still incompletely understood, but they include using variations of the homologous end joining reactions as shown in figure 553. You could just have a look at the figure right now. This is the figure. Please take a screenshot if you think it's helping you, right? To restart replication forks that have run into a break in the parental DNA template. General recombination is also essential for the accurate chromosome segregation that occurs during meiosis in fungi, plants, and animals. The crossing over of chromosomes that results causes bits of genetic information to be exchanged to create new combinations of DNA sequences in each chromosome. The evolutionary benefit of this type of gene mixing is apparently so great that the reassortment of genes by general recombination is not confined to multicellular organisms. It is also widespread in single-cell organisms. The central features that lie at the heart of the general recombination mechanism seem to be the same in all organisms. Most of what we know about the biochemistry of genetic recombination was originally derived from studies of bacteria, especially of E. coli and its viruses, as well as from experiments with simple eukaryotes such as yeast. For these organisms with short generation times and relatively small genomes, it was possible to isolate a large set of mutants with defects in their recombination processes. The identification of the protein altered in each mutant then allowed the collection of proteins that catalyze general recombination to be identified and characterized. More recently, close relatives of these proteins have been discovered, and extensively characterized in Drosophila, mice, and humans as well. General recombination is guided by base-pairing interactions between two homologous DNA molecules. The abundant general recombination observed in meiosis has the following characteristics. 1. Two homologous DNA molecules that were originally part of different chromosomes cross over, that is, their double helices break, and the two broken ends join to their opposite partners to reform two intact double helices, each composed of parts of the two initial DNA molecule. Right, that shows on the figure here. This is the figure. Please take a screenshot if you may. All right, proceeding. The site of exchange, that is where a red double helix is joined to a green double helix in 554. This is the other one. Can occur anywhere in the homologous nucleotide sequences of the two participating DNA molecules. At the site of exchange, a strand of DNA molecule has become base paired to a strand of the second DNA molecule to create a heteroduplex joint that links the two double helices in figure 555. So that's the one. Take a screenshot there. This heteroduplex region can be thousands of base pairs long. We explain later how it forms. No nucleotide sequences are altered at the site of exchange. Some DNA replication usually takes place, but the cleavage and rejoining events occur so precisely that not a single nucleotide is lost or gained. Despite its precision, general recombination creates DNA molecules of novel sequence. The heteroduplex joint can tolerate a small number of mismatched base pairs, and more importantly, the two DNA molecules that cross over are usually not exactly the same on either side of the joint. As a result, no recombinant DNA molecules, recombinant chromosomes, are generated. Figure 554 here is about the general recombination in detail. So the breaking and rejoining of two homologous DNA double helices creates two DNA molecules that have crossed over in meiosis. This process causes each chromosome in a germ cell to contain a mixture of maternally and paternally inherited genes. Right? And we're back. Similarly, a heteroduplex joint. This structure unites two DNA molecules where they have crossed over. Such a joint is often thousands of nucleotides long. The mechanism of general recombination ensures that two DNA double helices undergo an exchange reaction only if they contain an extensive region of sequence similarity, homology. The formation of a long heteroduplex joint requires such homology because it involves a long region of complementary base pairing between a strand from one of the two original double helices and the complementary strand from the other double helix. But how does this heteroduplex joint rise, and how do the joint two homologous regions of DNA at the site of crossing over recognize each other? As we shall see, recognition takes place during a process called DNA synapses, in which base pairs form between complementary strands from the two DNA molecules. This base pairing is then extended to guide the general recombination process, allowing it to occur only between DNA molecules that contain long regions of matching or nearly matching DNA sequence. Next will be meiotic recombination, is initiated by double-strand DNA breaks. The mechanism of general recombination ensures that two DNA double helices undergo an exchange reaction only if they contain an extensive region of sequence similarity, homology. The formation of a long heteroduplex joint requires such homology because it involves a long region of complementary base pairing between a strand from one of the two original double helices and a complementary strand from the other double helix. But how does this heteroduplex joint arise, and how do the two homologous regions of DNA at the site of crossing over recognize each other? As we shall see, recognition takes place during a process called DNA synapses, in which base pairs form between complementary strands from the two DNA molecules. This base pairing is then extended to guide the general recombination process, allowing it to occur only between DNA molecules that contain long regions of matching or nearly matching DNA sequence. Meiotic recombination is initiated by double-strand DNA breaks. Now, the extensive base pair interactions cannot occur between two intact DNA double helices. Thus, the DNA synapses that is critical for general recombination in meiosis can begin only after a DNA strand from one DNA helix has been exposed and its nucleotides have been made available for pairing with another DNA helix. In the absence of direct experimental evidence, theoretical models were proposed based on the idea that a break needed to be made in just one of the two strands of a DNA helix to produce the exposed DNA strand required for DNA synapses. This break in the phosphodiester backbone was thought to allow one of the nicked strand ends to separate from its base paired partner strand, freeing it to form a short heteroduplex with a short intact DNA helix, thereby beginning synapses. Models of this type are reasonable in theory, and they have been described in textbooks for nearly 3 decades. In the early 90s, sensitive biochemical techniques became available for determining the actual structure of the recombination intermediates that form in yeast chromosomes at various stages of meiosis. These studies reveal that general recombination is initiated by a special endonuclease that simultaneously cuts both strands of the double helix, creating a complete break in the DNA molecule. The 5' end at the break are then shooed back by an exonuclease, creating protruding single-stranded 3' ends. It is these single strands that search for a homologous DNA helix with which to pair, leading to the formation of a joint molecule between a maternal and a paternal chromosome, as shown in the figure here. See that there? So please take a screenshot of that. It should be helpful. And now the same details here. General recombination in meiosis. As indicated, the process begins when an endonuclease makes a double-strand break in a chromosome. An exonuclease then creates two protruding 3' single-stranded ends, which find the homologous region of a second chromosome to begin DNA synapses. The adjoint molecule formed can eventually be resolved by selective strand cuts to produce two chromosomes that have crossed over, as is shown. You see this? Right. And on to the subject matter again. In the next section, we begin to explain how a DNA single strand can find a homologous double-stranded DNA molecule to begin DNA synapses. The next heading will be DNA hybridization reactions provide a simple model for the base pairing steps in general recombination. In its simplest form, the type of base pairing interaction central to the synapsis step of general recombination can be mimicked in a test tube by allowing a DNA double helix to reform from its separated single strands. This process, called DNA renaturation or hybridization, occurs when a rare random collision juxtaposes complementary nucleotide sequences on two matching DNA single strands, allowing the formation of a short stretch of double helix between them. This relatively slow helix nucleation step is followed by a very rapid zippering step, as the region of double helix is extended to maximize the number of base pairing interactions, as shown in the figure. Right? Non-pairing interactions, pairing interactions. As you should take a screenshot and we look at these. This is a DNA hybridization. DNA double helices reform from their separated strands in a reaction that depends on the random collision of two complementary DNA strands. The vast majority of such collisions are not productive, as shown on the left, but a few results in a short region where complementary base pairs have formed helix nucleation. A rapid zippering from them leads to the formation of a complex double helix. Through this trial and error process, a DNA strand will find its complementary partner even in the midst of millions of non-matching DNA strands. A related, highly efficient trial and error recognition of a complementary partner DNA sequence seems to initiate all general recombination events. Formation of a new double helix in this way requires that the annealing strands be in an open, unfolded conformation. For this reason, in vitro hybridization reactions are performed at either high temperatures or in the presence of an organic solvent such as formamide. This condition melts out the short hairpin helices that result from the base-bearing interactions that occur within a single strand that folds back on itself. Most cells cannot survive such harsh conditions and instead use a single-strand DNA-binding SSB protein to melt out the hairpin helices and help anneal their complementary single strands. This protein is essential for DNA replication, as described earlier, as well as for general recombination. It binds tightly and cooperatively to the sugar phosphate backbone of all single-stranded DNA regions of DNA, holding them in an extended conformation with the bases exposed. This is a picture of that. DNA polymerase. Cooperating protein binding strategies, region of chain. So in the single strand can base pair efficiently, either with a nucleoside triphosphate molecule in DNA replication or with a complementary section of another DNA single strand as part of a genetic recombination process. The partner that a DNA single strand needs to find in the synapsis step of general recombination is a DNA double helix rather than a single strand of DNA, which is shown in the figure 556. Right? In the next section, we see how the critical event that allows DNA hybridization to begin during recombination, the initial invasion of a single strand of DNA into a double helix, is achieved by the cell. The REC-A protein and its homologs enable a DNA single strand to pair with a homologous region of DNA double helix. General recombination is more complex than the simple hybridization reactions just described, involving single-stranded DNA, and it requires several types of specialized proteins. In particular, the E. coli REC-A protein has a central role in the recombination between chromosomes. It and its homologs in yeast, mice, and humans make synapses possible. What you're about to see is the structure of the REC-A and RAD51 protein DNA filaments. The RAD51 protein bound to a DNA single strand, RAD51, is a human homolog of the bacterial REC-A protein. 3 successive monomers in this helical filament are colored, as you will be seeing more of it here now. This, what you see, is the detail of the picture there. Now a short section of the REC-A filament with the 3D structure of the protein fitted to the image of the filament determined by electron microscopy. The 2 DNA protein filaments appear to be quite similar. There are about 6 REC-A monomers per turn of the helix, holding 18 nucleotides of single-stranded DNA that is stretched out by the protein. The exact path of the DNA in this structure is not known, courtesy of Edward Eagleman, B. from XYU et al., Sheer Molecular Biology, 1998. Thank you. Like a single-strand DNA binding protein, the REC-A type of protein binds tightly and in long cooperative clusters to single-stranded DNA to form a nucleoprotein filament. Because each REC-A monomer has more than one DNA binding site, a REC-A filament can hold a single strand and a double helix together. This allows it to catalyze a multi-step DNA synapses reaction between a DNA double helix and a homologous region of single-stranded DNA. The region of homology is identified before the duplex DNA target has been opened up through a three-stranded intermediate in which the DNA single strand forms transition base pairs with bases that flip out from the helix and the major groove of the double-stranded DNA molecule. This reaction begins the pairing shown previously in the figure 55-56 we've already seen, and thereby initiates the exchange of strands between two recombining DNA helixes. The DNA synapse catalyzed by the Nacw protein. In vitro experiment shows that several types of complex are formed between a DNA single strand and covered with a Nacw protein, red in color, and DNA double helix, green in color. First, a non-base paired complex is formed, which is seen in the diagram here. You see that there? And which is convenient through transient-based, converted through transient base flipping, as on the former figure, to a three-stranded structure as soon as a region of homologous sequence is found. This complex is unstable because it involves an unusual form of DNA and it spins around out a DNA heteroduplex, one stranded green and the other strand red, plus a displaced single strand from the original helix green. Thus, the structure shown to this diagram migrates to the left, reeling in the input DNAs while producing the output DNAs. The net result is a DNA strand exchange identical to that diagram earlier, right? Courtesy is the West Biochem of 1992. Now, once DNA synapses has occurred, the heteroduplex region where the strands from two different DNA molecules have begun to pair is enlarged through a process called branch migration. Branch migration can take place at any point where two single-strand DNAs or single DNA strands with the same sequence are attempting to pair with the same complementary strand. In this reaction, an unpaired region of one of the single strands displaces a paired region of the other single strand, moving the branch point without changing in the total number of DNA base pairs. Although spontaneous branch migration can occur, it proceeds equally in both directions, so it makes little progress and is unlikely to complete recombination efficiently. The Nacw protein catalyzes an unidirectional branch migration, readily producing a region of heteroduplex DNA that is thousands of base pairs long. Two base pairs. So two types of DNA branch migration observed in the experiment in vitro. Spontaneous branch migration is a back and forth, random walk process, and it therefore makes little progress over long distances. Nacw protein directed branch migration proceeds. And we'll see the detail on the figure there. At a uniform rate in one direction and may be driven by the polarized assembly of the Nacw protein filament on a DNA single strand, which occurs in the direction indicated. Special DNA helicases that catalyze branch migration even more efficiently are also involved in the recombination. For that example, you need to see figure 63. Go on, take a screenshot. That should be it. Now, the catalysis of directional branch migration depends on a further property of the Nacw protein. In addition to having two DNA binding sites, the Nacw protein is a DNA-dependent ATPase with an additional site for binding and hydrolyzing ATP. The protein associates much more tightly with DNA when it has ATP bound than when it was ADP bound. Moreover, new Nacw molecules with ATP bound are preferentially added at one end of the Nacw protein filament, and the ATP is then hydrolyzed to ADP. The Nacw protein filaments that form on DNA may therefore share some of the dynamic properties displayed by the cytoskeletal filaments formed from actin or tubulin, as seen in chapter 6, 16. An ability of the protein to treadmill and unidirectionally along a DNA strand, for example, could drive the branch migration reaction. There are multiple homologs of the RECA protein in eukaryotes, each specialized for a special function. When one compares the proteins that catalyze the basic genetic functions in eukaryotes with those in bacteria such as E. coli, one generally finds that evolutionarily related proteins are present that catalyze similar reactions. In many cases, however, multiple eukaryotic homologs take the place of a particular bacterial protein, each specialized for a specific aspect of the bacterial protein's function. The generalization applies to the E. coli RECA protein. Humans and mice can contain at least 7 RECA homologs. Each homolog is presumed to have special catalytic activities and its own set of accessory proteins. The RAT51 protein is a particularly important RECA homolog in yeast, mice, and humans. It catalyzes a synaptic reaction between a DNA single strand and a DNA double helix in experiments in vitro. Genetic studies in which the RAT51 protein is mutated suggest that this protein is critical for the health of all 3 organisms. When required to repair replication forks that have been accidentally broken down during the normal course of each S phase. Its proper function requires multiple accessory proteins. 2 of these, the BRCA1 and BRCA2 proteins, were first discovered because mutations in their genes are inherited in a subset of human families with a greatly increased frequency of breast cancer, whereas the removal of the RAT51 protein kills a cell. Less drastic changes in its function caused by an alteration in such an accessory protein is thought to lead to an accumulation of DNA damage that often, in a small proportion of cells, gives rise to a cancer. Different RECA homologs in eukaryotes are specialized for meiosis or for other unique types of DNA synaptic events that are less well understood. It is likely that each eukaryotic RECA homolog loads onto a DNA single strand to begin a general recombination event only when a particular DNA structure or cell condition allows a protein to bind there. General recombination involves a holiday junction. It's holiday junction. The synapses that exchanges the first single strand between 2 different DNA double helices are presumed to be the slow and difficult step in a general recombination event. After this step, extending the region of pairing and establishing further strand exchanges between the 2 DNA helices is thought to proceed rapidly. In most cases, a key recombination intermediate, the holiday junction, also called a cross-strand exchange, forms as a result. In a holiday junction, let's see what a holiday junction is. It's basically referred in the glossary of the National Library of Medicine. A holiday junction is an X-shaped structure observed in DNA undergoing recombination in which the 2 DNA molecules are held together at the site of crossing over, also called as cross-strand exchange. In a holiday junction, the 2 homologous DNA helices that have initially paired are held together by the reciprocal exchange of 2 of the 4 strands present. When originating from each of the helices, as shown in figure 561a, a holiday junction can be considered to contain 2 pairs of strands. Let's see the figure. Right, so this is the figure. A holiday junction can be considered to contain 2 pairs of strands, 1 pair of crossing strand and 1 pair of non-crossing strands. The structure can isomerize, however, by undergoing a series of rotational movements catalyzed by specialized proteins to form a more open structure in which both pairs of strands occupy equivalent positions, figures 561 and 62. This structure can in turn isomerize to a conformation that closely resembles the original junction, except that the crossing strands have been converted into non-crossing strands and vice versa. A holiday junction and its isomerization as described in this text. The synapsis step is, in general, recombination is catalyzed by a rec-A type protein bound to a DNA single strand. This step is often followed by a reciprocal exchange of strands between two DNA double helices that have thereby paired with each other. This exchange produces a unique DNA structure known as holiday junction, named after the scientist who first proposed its formation. The initially formed structure contains two crossings inside strands and two non-crossing outside strands. An isomerization of the holiday junction produces an open symmetrical structure. Further isomerization can interconvert the crossing and non-crossing strands, producing a structure that is otherwise the same as that in A. Electron micrograph of a holiday junction. This view of the junction corresponds to an open structure illustrated in figure 561b, courtesy of Huntington, Potter, and David Dressler. Once the holiday junction has formed an open structure, a special set of proteins can engage with the junctions. One of these proteins uses the energy of ATP hydrolysis to move the crossover point, the point at which the two DNA helices are joined rapidly along the two helices, extending the region of the heteroduplex. Figure 563 shows enzyme-catalyzed double-branch migration at a holiday junction. An E. coli tetramer of the RuV-A protein, green, and two hexamers of the RuV-B protein, pale gray, bind to open form of the junction. The RuV-B protein uses the energy of ATP hydrolysis. To move the crossover point rapidly along the paired DNA helices, extending the heteroduplex region as shown. There is evidence that similar proteins perform this function in vertebrate cells, courtesy SC West, 1998. To regenerate two separate DNA helices and thus end the exchange process, the strands connecting the two helices in a holiday junction must eventually be cut, a process referred to as resolution. There are two ways in which a holiday junction can be resolved. In one, the original pair of crossing strands is cut. The invading or inside strands in figure 561a, which we've already seen, I guess. In this case, the two original DNA helices separate from each other nearly unaltered, exchanging the single-stranded DNA that formed the heteroduplex. In the other way, the original pair of non-crossing strands is cut, the inside strands in figure 561. Now the outcome is far more profound. Two recombinant DNA chromosomes are formed, having reciprocally exchanged major segments of double-stranded DNA with each other through a crossover event. The resolution of a holiday junction to produce crossed over chromosomes. In this example, homologous regions of a red and green chromosome have formed a holiday junction by exchanging two strands. Cutting these two strands would terminate the exchange. Right, without crossing over, with isomerization of holiday junction steps A, B, and C, the original non-crossing strands become the two crossing strands. Cutting them now creates DNA molecules that have crossed over bottom. This type of isomerization may be involved in the breaking and rejoining of two homologous DNA double helices in the meiotic general recombination. The gray bars in the central panels have been drawn to make it clear that the isomerization events shown can occur without disturbing the rest of the two chromosomes. The general analysis revealed that the heteroduplex region of several thousand base pairs are readily formed during recombination. as described next. The process of these heteroduplexes, which generally consist of nearly identical paired complementary strands, can further change the information in each resulting DNA helix. General recombination can cause gene conversion in sexually reproducing organisms. It is fundamental law of genetics that each parent makes an equal genetic contribution to an offspring, which inherits one complete set of genes from the father and one complete set of genes from the mother. Thus, when a diploid cell undergoes meiosis to produce 4 haploid cells discussed in chapter 20, exactly half of the genes in these cells should be maternal genes that the diploid cell inherited from its father, and the other half paternal genes that the diploid cell inherited from its father. In some organisms, fungi for example, it is possible to recover and analyze all 4 of the haploid gametes produced from a single cell by meiosis. Studies in such organisms have revealed rare cases in which the standard rules of genetics have been violated. Occasionally, for example, meiosis yields 3 copies of the maternal version of the gene and only 1 copy of the paternal allele. Alternative versions of the same gene are called alleles. Alleles would be in the glossary. The explanation for that is the definition is one of a set of alternative forms of a gene. In a diploid cell, each gene will have 2 alleles, each occupying the same position locus on a homologous chromosomes. So these are alternative forms of a gene. All right. And this phenomenon is known as gene conversion. Gene conversion. Right? Gene conversion often occurs in the association with homologous genetic recombination events in meiosis, more rarely in mitosis, and it is believed to be a straightforward consequence of the mechanism of general recombination and DNA repair. Genetic studies show that only small sections of DNA typically undergo gene conversion, and in many cases only a part of a gene is exchanged. 565, figure 565, gene conversion in meiosis, as described in chapter 20. Meiosis is the process through which a diploid cell gives rise to 4 haploid cells, germ cells, eggs, and sperm, for example. Now, in the process of gene conversion, DNA sequence information is transferred from the one DNA helix that remains unchanged, a donor sequence, to another DNA helix whose sequence is altered and accept the sequence. There are several different ways that might happen, all of which involve the following 2 processes. 1, a homologous recombination event that juxtaposes 2 homologous DNA double helices, and 2, a limited amount of localized DNA synthesis, which is necessary to create an extra copy of one allele. In the simplest case, a general recombination process forms a heteroduplex joint, of which the 2 paired DNA strands are not identical. See figure 555. Right, you see that? In which the 2 paired DNA strands are not identical in sequence and therefore contain some mismatched base pairs. If the mispaired nucleotides at one of the 2 strands are recognized and removed by the DNA repair enzymes that catalyze mismatch repair, an extra copy of the DNA sequence on the opposite strand is produced. That's in 566, as shown below. The same gene conversion process can occur without crossover events, since it simply requires that a single DNA strand invade a double helix to form a short heteroduplex region. The latter type of gene conversion is thought to be responsible for the unusually facile transfer of genetic information that is often observed between the different gene copies in a tandem array of repeated genes. Now, gene conversion is shown here in this diagram. In this process, heteroduplex joins are formed at the sites of the crossing over between homologous material and paternal chromosomes. If the maternal and paternal DNA sequences are slightly different, the heteroduplex joint will include some mismatched base pairs. The resulting mismatch is in the double helix may then be corrected by the DNA mismatch repair machinery, which can erase nucleotides in either the paternal or the maternal strand. The consequence of this mismatch repair is gene conversion, detected as a deviation from the segregation of equal copies of maternal and paternal alleles that normally occurs in meiosis. And the next portion would be genetic recombination events have different preferred outcomes in mitotic and meiotic cells. I'll just give a general mention of this because this lecture would be over in a minute or two from now, and it would be continued in the next lecture. All right. So basically, this is something that we have seen in meiotic recombination. It starts with a very bold stroke, the breakage of both strands of the double helix in one of the recombining chromosomes. So how does the meiotic process that follows differ from the mechanism, also based on general recombination that cells use for the precise repair of the accidental double strand breaks that occur in chromosomes? The homologous end joining reaction, figure 553, you see from here. This is the figure there, right? And now, in both cases, the two new chromosomes ends produced by a double strand break are subjected to a degradative process, which exposes a single strand with an overhanging 3' end. Moreover, in both cases, this strand seeks out a region of unbroken DNA double helix with the same nucleotide sequence and undergoes a synaptic reaction, which with it that is catalyzed by the REC-A type of protein. For double strand DNA break repair, DNA synthesis extends the invading 3' end by thousands of nucleotides using one of the strands of the recipient DNA helix as a template. If the second broken end becomes similarly engaged in the synaptic reaction, a John molecule will be formed as seen in 556. Depending on subsequent events, the final outcome can either be restoration of the two original DNA helices with repair of the double strand break, the predominant reaction in mitotic cells, a crossover event that leaves heteroduplex joints holding two different DNA helices together. The predominant reaction in meiotic cells. So that ends the chapter here. The short introduction here. It is thought that the crossover events are set by specific proteins that guide these reactions undergoing cell meiosis. I'll see you in the next lecture shortly.